Hello, I'm Katie Butts-Dehm, your instructor for Bio 110. Tonight we're going to work on the cell lab. And so make sure that you have the cell lab handout, follow the instructions, and the cell lab answer sheet so you can write everything down, draw pictures, things like that. And it's going to be the cell lab answer sheet that you'll want to turn in to me. Let's see. So I'm going to go ahead and share the screen just to make sure I can and make sure there's sound. And here's the screen. And now you can see a few of the documents. Here's the handout. Make that a little bigger. And there's some videos that we're going to look at. And actually, you should go watch these videos on your own. This Carson flip microscope really shows you how to use the high power pocket microscope, microflip microscope, and it's 100 to 250x because it'll show you the image on the microscope slide will be 100 times the size or 250 times the size. And then there's videos and how to prepare wet mount slides. So make sure that you look at all of these URLs before you start, especially this first one, so you'll know what you're doing with that. And then the answer sheet that you have is right. That's not it. Here it is. So this is what you're going to submit to me. Now you can handwrite on this and scan it in your computer and submit it that way into the assignment, or you can type it in, okay? But either way, that's what you're going to turn in to me. So make sure you have all the supplies first, and let's see, I'll stop sharing the screen just so we can look. And again, I have my list here. So you should have your flip microscope, pocket microscope, which is right here. And you should also have, let's see, a letter E slide, which, oh, mine is being used, it has a letter E on it. You should have, I don't know why this looks out of focus. It didn't earlier. Let's see. You should have a bacteria slide, which might say bacteria on it, and it'll be, you know, you can see a little purple stain, so you can see the bacteria. You should have 4 blank microscope slides, and you can reuse these slides, just clean them off when you're done. And you need to also have 4 cover slips. The cover slips are plastic, and there may or may not be tissue paper separating them, and they're clear. And then you need a methylene blue stain, which it should be in a little dressing cup, maybe in a little bottle. And then it's labeled methylene blue stain. Well, really not focusing very good, but methylene blue stain, you can read it on your own bottle. And then a packet of yeast, and I believe this is optional. I put my yeast in some warm water, just a little bit of warm water and yeast. And that's if you want to look at yeast cells. You also need a flat toothpick or a spoon. A flat toothpick is better, but I have a spoon and I have a butter knife. Now, make sure that you don't have, if you use something like this, nothing with serrations, because we're going to get cells from the inside of our mouth. And you don't want to cut it. You don't want blood cells. You want cheek cells. So the flat end of a flat toothpick is what we usually use at school. But I didn't think that you would want me touching all of your individual toothpicks and sending them to you. And, you know, maybe I could pick them out with the tweezers or something in the future. But a piece of onion. So I have some onion here, little pieces, and a thin piece of lettuce, a small knife, which I'll need to get, and some water, okay? So that's what you need. The introduction will give you some background material on cells. And we're going to be looking at plant cells, at animal cells, at bacteria cells. And the bacteria cells, you'll just barely be able to see the bacteria. But also letter E slide. And then, let's see, and some fungi cells or yeast cells. Okay, so the first thing we're going to do is the letter E slide. So you're going to use the microflip pocket microscope. And I don't know how this is going to work. I put my cell phone on hoping I could show you the image of it, but maybe not. So the first thing that you have is your microscope comes with this little clip. And you need to figure out which side, like there's circles on my cell phone, which one works with your camera. And I know for mine, it's the middle one with my regular camera. So you want to put this clip where the hole will go on, let's see, one side. And let's see, when I have my camera turned on, so maybe I won't be able to be in the Zoom meeting and show you it. I don't know. But when I have my camera turned on, oh, I believe it's this way. Then this goes over it, so like that. And then I should be able to see, you can sort of see that there's a circle showing around. And then what you're going to want to do, so that clip is on and it's on pretty good. So then I want to go ahead and turn this on and make sure, for me, this is down. So I want to put a slide in there. And I just click it. Oh no, it just worked. There, okay. So the light turned on and that's there. And then this will go right onto the cell phone. I wish I had something for the cell phone. This hooks right in here. So you see a circle now in your phone. I don't know if you can see that. It looks like the reflection of the light, but there's a circle. I have to slant it though, like that. Then you're going to go ahead and let's make sure you can see this. So I have the circle, slide the E slide right under there. Until you can see it. And right now I know something's off, maybe with the clip there, that's better. So I have a round circle that's lit and we're going to just slide this E until you see it. Oh, wait. Also, I want to make sure this is turned to 100, not 250. So up there, that's 100, not 250. For some reason, I saw this right before this video, and I was able to see the E. Now I am not. Seeing this. That's the right one. Clip's not on right. Okay, now the clip is on right. There. Okay. So now I can see the E, and then I focus in on it, and there it is. And the deal about this is it's so large that the E is so close up that you can't see the entire E on here at 100x. But you're going to find the E also, there's little dye crystals in here. And to me, it's strange because I actually saw the entire E earlier. So maybe if no, it's not going to be that one, it's the other one. That's pulled all the way back. And this is the focus. I'm going to do this one, reset it one more time. And don't feel bad if you have to reset it because, well, I had to. And again, I did it perfect first time right before I logged onto this. But I'm recording the video now, so it's not going to be perfect anymore. There. Okay. And then there's the E. Oh, but of course, it's such a high magnification. That's perfect. And that's tiny. So anyway, what had happened, I was having issues because the circle got really small and I just had to enlarge it to where I could see a little bit more. But I think you can actually see the E that I just had on the screen and moved it away. But the little purple, little blue dots I have are around the E. Now I had to use the slide I had last semester. So I believe it's a little bit different than the one this semester. But there you go. And you can, once you see the E, I'm going to enlarge this a little bit more too, and find it again. There, you see it? Then you can focus on the E. A lot less control with this than you would have in a microscope in the lab, but there it is. There's the E, and you can take a picture, and there you go. And I can tell when that little circle or the little stain that looked like it was on the top, that was because my flip moved just a tiny bit. I'll tell you, this would be a lot better. Oh, look, my whole entire E shows. And I can take a picture. I wonder what this means, switch to macro. Switch to macro. I wonder what that'll do. Oh, it doesn't do anything. I don't even know how to unswitch. Okay, oh, look, it's tiny. Can you see that? And I can just blow up my screen and I can see it larger. And there's my E. That looks great. That really looks good. So I'm going to go ahead and take this slide out. And I think what I'd like to do is look at the bacteria slide. And I'm just going to look at that now since I have this all pulled up. And bacteria are going to be really, really, really small. So it's going to be hard to see them. And at least you can look at the purple and that might help. I see a bunch of skin cells on there. And I don't know why it looks like an E, but there's no E there. Okay, get rid of the little stain. And now I want to put the magnification all the way up. Oh, I was doing focus there. the magnification all the way up and up there. And then I'm going to go ahead and focus using, you turn this for the focus. Okay. Okay, so in here, I see some things that look like cereal flakes. And the cereal flakes are basically skin cells. From people handling the slide, like myself, they're going to be all over. So you can move this around. And if you see little dots, like there's a big purple blob there, that's mostly just the stain. I wonder if I put this right above it, if it would show you anything or not. It doesn't really look the right color, but so I'm going to move the slide around, and there's little dots. Actually, I think you can see it better from back here, a little bit better from back here. But once you find the little dots and you get focused on them, and they should be stained purple, and this is about the biggest bacteria that we can get. Oh, that's going to be better. So, and it's Bacillus megaterium, I believe, and it's the largest bacteria with single cells that we have. So once you get that, then you are going to focus on it, take a picture, and save it. And then the bacteria that you have, I wonder if I could show it in the Zoom meeting in the other one. I don't think so. No, but anyway, once you have it, click it, save a picture of it. Oh, that's really nice. Let's see if I can enlarge it. I can see it actually a lot better when I enlarge my camera screen, and then I can focus some more. I'm surprised. And oh, wow. And then I can actually see some singles, but just barely. That's the thing about this, with the size and the magnification. Most bacteria you look at are really high magnification, like 1000x, and this goes up to 250. So there you go. I saw some right near the big stain glob, right on the edge of it. There's some. Yeah, and they're bacillus. They're singles. Okay, so I'm done with that one. Let's see. So the next thing we're going to do is, well, I'm going to check real quick. I'll turn this off and look at my cell feed. Oh my goodness, what did I do? There it is. Okay. Let's see. So we did the E slide and the bacterial slide, and we're going to either draw it or put it on, take a picture of it. And you can insert your images into the lab, or I found out sometimes Brightspace is cutting the images off if they're in the middle or moving them around. If that's a problem, you can just insert the images separately. It would be nice if they were all on one page though, but anyway, they don't have to be. So your cheek cell smear, that's the next thing that you are going to do. And let's see. So we did introduction, the E slide, the bacterial smear, and you know, for the bacteria smear, you just basically smear it in there, put some stain over it, rinse. Well, you smear it on there, you have to let it dry, then you heat fix it, then you put some stain on it. And it's not a gram stain, it's just a regular like purple stain. I'm not sure what they used. But then after that, you let it sit for however long the stain says, usually like a minute, and then you rinse that off and you blot it on some Builder's paper, and there you go. There's your bacteria. The gram stain is more complex, but so for the cheek smear, here's the directions. You're going to put a drop of methylene blue on a glass slide. Normally you use the flat end of the toothpick, but we're not home, so spoon or or I'm using a butter knife with no serrations. Okay, don't use anything that will hurt you. But you're going to rub that against the inside of your mouth, and then you're going to stir your cheek cells into methylene blue drop. Now, usually when I'm in the lab, I'll dilute some methylene blue with a little bit of water, and you know, but you have straight methylene blue. So if you want to mix half and half with water, it might be helpful. Most people don't dilute it, but you're going to hold. This is a wet mount slide. This is why I pulled this up, because I want you to see this. You're going to take the slide. You're going to put your drop of methylene blue. You're going to stir your cheek cells in that. And then you're going to take the cover slip and you gently lower it over that drop. And that's how you make a wet mount slide. You don't put it flat, because putting it flat will leave a bunch of air bubbles, and the air bubbles look like big tires. So that's what we're going to do next. Stop share. Yeah. And then switch camera. Oh no. Switch camera. There we go. So here's the switch camera, and the slide and the bacteria slide. So now I need a blank slide, and I need the plain blue. And you know what? What I'm going to do is I'm going to dilute a little bit, because you can see it better. Oh, no. It won't come up. Oh, but there, it came open. I just feel like you can see it better. Like I said, other people don't dilute it, but I do. And it just gets so dark. And I'm just going to take one of my droppers from before that I rinsed off. And there is some diluted methylene blue. Maybe I diluted a little too much. We'll find out. And then here's my slide. And by the way, oh, I'm sorry, I didn't realize my video stopped. So I put a drop of methylene blue, and I put some water into it using this. And actually, I think I put more water than I really wanted. So here's my dilute methylene blue that I'm going to use. And again, you can use a straight out methylene blue. And there, this just had water in it. So, okay. Now I have the slide here. Oh, and you're going to need to see this. So what I'm going to do is I'm taking this, no serrations, gently rub it. You don't need blood. Don't get blood. Well, and you're going to have this like gross, nasty looking film. And then, oh, I wish I had the drop of methylene blue already on there. Now I'm going to do put my drop of methylene blue already on there. And then I'm going to rub this in my film in there. And this film is weird. Maybe I should have taken this. Well, see, normally you just stir a little toothpick in there. You stir it in the drop, but this is a mess. We'll see. I'm not going to use that knife again. Oh, you can see that. Here. I will. Okay, so here it is right here. It's a big old drop, way bigger than you would normally get. To get that knife, like the knife had the slide, you know, the stuff on it, and there was no way I could get that to go on to the slide. So I just sucked up that drop, put it on that knife and stirred it around and then put it back on there. So it's a little bit of a mess, but we'll see. If it doesn't work, it's not the end of the world. If it looks bad, I can just redo it. You know, so, and the slides and the cover slips, you can wash them and reuse them. So you slide that till it touches the drop and the drop spreads out on it. And then it's at an angle, and then you gently lower it. And there it is. Now I can see some air bubbles. So I know that there's some air bubbles. And I'm going to be looking, they're going to look like tires. But it's weird because around the air bubbles, they have, what is it? like it's like a big black tire. And everyone's like, oh, look, I found something. Now mine, I had a little bit too much liquid that I don't want my cover slip floating. So I just am taking just the flat edge of a paper towel and putting it down there, and it sucks some of the liquid out. And there we go. So first I'm going to put this at 100x. Now, too bad I'm not very good at just sticking my cell phone in. Every single time I have to look at my cell phone, I have to make sure that the opening is over the camera that works. I can't just shove my cell phone in there. It doesn't work for me, okay? And then I have to make sure that I turn on the camera and then put this on here. And look, it's got that little tiny circle, so I'll have to enlarge it. And then it's got the little stain looking thing, so I have to move this to get it perfect. And turn on the light, so it's a lit up circle. I have no idea why it's all blue right now. I mean, what's it pointing at that it's all blue? Oh, yeah, I don't know, maybe it's not. Let's see if I go that way. Oh, that's the ceiling. Anyway. So here this is, oh, look, I can see cells already. So it did work. Oh, look at all those cells. Oh my goodness, that's perfect. And it's perfect. Oh wait, now it doesn't look as good. Oh, there, oh. Now one of those cells is perfect. Look, okay, stay focused. I move it around, it goes out of focus. So I'm gonna go and do that. So you can't see it now. So hopefully these images I'm taking, then I'll show you because I'm saving them. And this, by the way, is the 100x. So these, I believe, I usually look at on the 40x, and it's 100x right now. That's a 40x light microscope at school. It is amazing that they make these microscopes. You can just hook onto your cell phone. It's just really amazing. But I'm going to turn the power up. Wow. And the thing is, if you turn, like you can see, even at where I had it 100, oh, wow. You can see the nucleus, you can see the cell membrane, you can see all parts of things. And again, the image that you are seeing on the screen is not nearly as good. Oh, I thought I did. I went by an air bubble real fast. But it's not, it looks way better in person. Way better in person than what you're seeing over there. I'm going to go even up a little bit more, and that's the highest magnification. So I believe, and I'd have to look at the sheet again, but I believe that you are supposed to draw a few cells. And if there's 1,000 cells on your screen, you don't need to draw 1,000. Draw like 5 or 6, and then you'll have to label the cell membrane. You'll have to label the nucleus and the cytoplasm, which is everything between the nucleus and the cell membrane, okay? And these are my cells, and I am an animal. Oh, there's, well, not really an air bubble, but it's got, like you can see, the whole air patch. But there's no cell wall in these because these are animal cells. But it's neat because everything in there, in that little nucleus, okay, that is all the blueprints to make another U. So the whole instructions to make another U is right there. And there's it's like thousands of cells in here just from rubbing that just a little bit, okay? So I think it's pretty neat, but there you go. There it is. Again, this doesn't even show, it's not even focused at all, and it's not even the right color. I really think the diluted methylene blue turned out a lot better than it would have had it not been diluted. So that's what I would do. So I'm going to take my slide, I'm going to set it aside, and later on I'll clean it, and I'll turn this off for now. So let's see, what do we need to do next? So again, insert the photo, or you can draw it, just draw it, do a drawing. Okay, so you can just do a drawing, label the cell membrane, the nucleus, and the cytoplasm in your cheek cell drawing. So now we're going to do onion skin, and we're also going to do a leaf or plant cell slide. So I brought just the end of some romaine lettuce. I don't know if it's going to turn out very good, but oh, well, okay. Yeah, the answer sheet has some more things. So what we're going to do is put a drop of methylene blue on a clean microscope slide. We're going to use a knife to cut a piece of onion, a small piece, and we're going to use a knife tip to remove the thin transparent layer on the inside. We'll put the onion skin on that with a drop of methylene blue and a cover slip, and we're going to view that. And then also, so that's a wet mount slide. The leaf one is a wet mount slide, but we're going to use water and not methylene blue. Okay. And then label any parts visible. Hopefully you'll be able to see a cell wall, the cytoplasm, maybe the nucleus, and maybe even some chloroplast. And then here's for the answer sheet. And I'm curious about that for now. But so let's see. Wait, stop share. So I got some pointy knives because normally I would use a scalpel for this. But if you don't have a scalpel at home, there you go. And, you know, I'm really surprised I don't need a scalpel. So when you have onion, then there's an inner, like really thin skin. Let's see, it may have came off on the other side of the onion because it's not coming off here. Let's try this part, this side. No, can't find it. See, I got all these pieces of onion. Oh, I see just a piece of it. It's really, really, really thin. Okay, really thin, and it's usually on an inside piece. Now, so there I'm going to put that really thin piece of onion right here. By the way, I am going to have some skin cells on this slide. So there's my really, really thin piece of onion. And you want to try to have it single layer because it's folded if you don't. Down, right down. And I'm going to get some new methylene blue. And it worked out so well. Down. Why that drop didn't fall off. I'm going to put some more, and it worked out so well being dilute that I'm going to dilute this one. Oh no. I wonder what happened to my rubber. There it is, right here. You know, I'm going to have to do a fresh one of these to put some water on it. And then I can get another one to mix it. Yeah, that should be plenty. About half and half. It really doesn't need to be that much. And then I'm going to put a drop of, now there's air bubbles all in it. Just put a drop of this on it. There we go. And now I'm going to get a cover slip. And try to just touch the cover slip by the edge so you don't get skin cells all over it. And I do have a big area of my slide with nothing on it. I don't know if you all could see that when I was doing it. So my thin piece of onion right on there, my drop of diluted methylene blue, and there's my cover slip. In that area, I don't like that area there, but what am I going to do about it? Maybe I could press this down a little bit. Oh yeah, there we go. So hopefully that'll look good. Onion is sort of on the edge. And I might as well just do this one. Scissors, I would have liked scissors a little bit better. And this is a little bit wilty. This is the thinnest part. So I'm going to take this leaf Scissors would have been better, just so I don't smash it. And I'm going to put a little drop of water, and so it'll still be a wet outside. I would get a smaller piece normally, but I don't want to smash it with my hands. Oh, there's scissors right there. Right here are scissors. Right, they were right over there. Well, there you go. And the cover slip. Definitely. There's a big, oh, look, you didn't see it again, did you, either? The leaf with a drop of water and then the cover slip over it. And there you go. The one other slide that I'm going to do, and this one is not on your assignment, but I thought it would be fun. If you have extra use from before, see. And if you have another cover slip, there's one, good, I have two of them. Right? Then I have some extra use. So I mixed a little bit with water, and there it is. It's been sitting bubbling up. And you know, I'm going to put just a little bit of the methylene blue in with it, because why not? And then I'm going to go ahead and put a cover slip. Here it is. There. No, no, no. You could see it, couldn't you? Okay. And then here's the cover slip right on it. There we go. So there's that slide of yeast is done. So we've got 3 slides to look at. You need to turn on your cell phone. Again, make sure your clip's on, where you've got the hole right there. And you may have to try again a few of them, but there's the hole in that. Oh, look, it wants me to download Candy Crush Saga. That's the last thing I need on this. Okay, and then we'll put this in here. Yep, and turn the light on. Whoa. Okay, enlarge the screen a little bit. And let's see how it enlarged itself. Oh, how strange. I have it backwards. I'm going to go at 100x, by the way. So I have to enlarge it a little more. And let's see. And then first, the onion was first on the list. So we're going to do the onion first. And you know what? I have to turn the camera around. Let's see so you can see what's going on. Okay. Yeah. I don't know that you can see any better. So I'm going to slide my slide into here. I told you there'd be lots of skin cells. Maybe you have the skin cell window. This is the onion one. You gotta find the onion. Right there. There it is. Onion. Oh, it looks great, by the way. I think it looked a little better where I was at before, but you definitely can see the cell walls. That's how it, oh, there. Definitely can see the cell walls. Oh, look, you can sort of see it from the video, but not as good as you could in real life. I don't know why it just wipes out the whole thing on the screen that you have. But I'm going to show you these images. But that's why I'm on Zoom on my phone. Now, some of them, they're overlapping. The cells are overlapping because it got folded. But I can just move over to a place where they're not overlapping. And then I'm going to turn this all the way up and then focus. Uh-oh, I moved it. And it was my nails. So I'll try to find that place again because it was so nice. That's one thing. If you have a microscope, you have knobs that you, oh, cool. That is really neat. You have knobs that you can move the slide with. So you don't have to worry about your slide being, moved a little bit by your hand moving. I don't even know what happened to it at this point, but I got some beautiful images. Oh, that's strange. I got some really nice images already. Oh, you know what? Maybe my clip moved or something like that, but I really got some nice pictures. So that one was the onion. And then the next one we're going to look at is the plant. And again, I've got to reset my clip. Be nice to just glue it on there. Oh, that's weird. I'm going to maybe, okay, it's there. Reset my clip. There's a circle. Put the clip in here. I don't know what all these settings it wants me to do. Turn it up. And then I'm going to put the plant. Right now I have it on such a high magnification. And I'm not sure about, oh, wow. Oh, okay. It's not in focus. You know, I do want to turn this to a lower magnification. Oh, neat. I'm going to turn it to a lot lower magnification. And then that, let's see. Looks like there's some leaf veins. I don't think I'm getting any individual cells showing on this plant. Okay. But it still is kind of neat. There's actually a type of algae. Oh, neat. Oh, good. That's better. That's maybe individual cells. Maybe I'll turn up that one. And I have to focus it. Again with me, yeah, it's way off. I wish I could. I'm supposed to stay. Put that there. Green, not focusing well without me moving it. There. Went right past it. That's a problem. Nope. Okay, and that's just such a thick part. It's not showing up good. Okay, there. So again, a lot thinner section of a plant would be better. So like seaweeds, there's a couple of seaweeds that are really good at looking at in the microscope. So this one's not, my romaine isn't quite as good, but I can see the veins. It's hard to see individual cells on it. Oh. I don't know why that won't let me enlarge this view. Another thing though that's kind of neat is really strange. But there we go. At least it's in focus. You can actually enlarge the picture on the computer sometimes if you don't have a nice picture. So there we go. And the last one that I'm going to look at is the yeast. And you know what I'm going to do is I'm actually going to reset, close and open my camera. I don't know what that is. Some news thing open. Oh, that's supposed to be my camera. Why does it want to show me commercials? I don't know. Okay, here, I'll make sure my camera's turned off there. Okay, now turn back on my camera. I'm just getting ads here as I'm doing this and put this back in the exact right place. Okay, I got a nice circle. It's in the right place. And I wonder what's going to happen if I throw the yeast in there first and just hope, because I keep moving the whole thing as I'm focusing it. Okay, put that right in here, and then hopefully it'll let me enlarge it. Oh, wow, it's moving around a lot, not because they are moving around, but because the water in there. And I'll tell you what I did. I put so much yeast in there that it is really difficult to see. And why is the magnification so low? I don't know. So I'm going to go around the edge, but I should have had a lot more water is what I should have, because they're just too tightly packed that they're hard to see. I'm still going to take a picture. And my slide looks like it's tightly packed throughout. And unfortunately, as I move it, it's moving the whole cover slip and getting dirty yeast everywhere. Maybe if I take this plastic thing and turn that, and I'm going to wipe this off, and I'm going to sanitize it later on. That maybe, because this you can put, look at that, see? It got these, if right on it and on here. And then I'm just going to set this down and see what I can see. And then this back. Okay. And then maybe if you get to the edge, when you have like too much or too thick of cells, then if you go toward the edge, sometimes you'll have less of them. But there we go. I have a picture of these. And for now, I'll show you the picture of these that I have in a minute. It's just not going to be that great, but that's okay. Okay, so what I'm going to do now, well, first I'm going to see, because I wonder if this will work, because I'm actually on the Zoom on my cell phone. And let's see. Anyway, oh, I wonder, this is going to be weird. Let's see. Okay. Hello. So what I'm going to do is see if I can show you these images on here. And I don't know if I can, but actually, let's see. I'm going to stop recording and figure out what I'm doing. Resume record. There, I'm back. And now I'm going to share the screen. And let's see. Images 1, 3, 2. I don't know where we want to start. Well, okay, so right here, these are some images that I took. These are the onion slides, and you'll notice you can see the cell membrane, cell wall really good. You can't see the cell membrane that good, but it's just inside the cell wall. You can see a nucleus over here and a nucleus over here. You can't see nuclei and all, that might be one there. But the cytoplasm is everything between the cell membrane and the nucleus. So plant cells have a thick cell wall of cellulose. So these are my cheek cells, and this one's a little out of focus. It's very large, but they're animal cells, so there's no cell wall. You can see the cell membrane, the nucleus, and everything between the nucleus and cytoplasm. So if you are going to draw yours, you don't need to draw like 50 cells or all of these. You can just draw like 2 or 3. Maybe there's this one's a nice one right up here with the cell membranes around the outside or plasma membrane, the nucleus in the middle, and then cytoplasm, everything between the nucleus and the cell membrane. So this one, it's a lower magnification, but I think it looks better because you can see all three parts that you would be able to see in this one, that one, that one. So notice no blood cells in there. This one's not as good. If I were to choose one to turn into meat, I would choose this one. And frankly, I could probably take the image and if I wanted to, and throw it on a PowerPoint. Oh, look, just happened to have a PowerPoint magically appear. And, oh, but that one's not shared with you. I just shared this one. But you could copy this, put it over to a PowerPoint, and then label it pretty easily. So let's see, I'm going to share a different one, new share. That was images 2. Here's images 3. Oh, the green line's not around it. So I'm going to go stop share. And then I'm going to go share screen again and images 3. It says I'm screen sharing, so I hope you can see this. There's just no line around it. So this is a bacteria. This one is not very good. It's not a great image, but there's little bitty rods. And like I told you, it's going to be hard to see them. So these are looks like the skin cells or epithelial cells are mixed in, but the little dots that you can barely see, those are the bacteria. You can barely see them. They're stained purple. Here's a big clump of something stained purple. And it looks like those are the only images I shared, 3 of the images. And again, they're not that great. Definitely I can see rods though in here. If I had, again, this is one that they are so small. And you normally look at them on oil immersion in a compound light microscope at 1000x. So this is 250x. And there's nothing really stabilizing or holding my setup still at that low magnification or that high magnification. We'll need it. This one turned out gorgeous. This is the first letter E that I did. Look at that. It's upside down and backwards too. The microscope turns everything upside down and backwards. But there's the letter E up close. And you can take a letter E or any letters out of newspaper print. Usually that's what we did in lab before they started selling them pre-made. Or these ones actually, the micro instructor made them. She prints off little labels, little stickers, prints them off and puts them on there. But there's a letter E. Oh, this is the one I did before class. These are the ones I did with you all. So isn't that neat? And then let's see, we're gonna stop sharing this one, and then I'm going to share, I don't think we looked at this one. Okay, so like I told you before, the yeast cells were so thick. I should have diluted that like 5 times with water because you really can't see anything at this dilution. And here is my romaine lettuce. You can see the like little veins in the lettuce. Unfortunately, close up, I didn't get a good picture. But look at this. Isn't that a nice 3D of that onion skin? You know, and do the best you can with them. And that looks like the same one from before. Do the best you can. You might have something like this. And if I took this image over, put it on a PowerPoint, then you never know. It might look pretty good. So let's see. I'm just going to share the screen and I will throw on a PowerPoint, which I have open. And I already had copied an image. I just copied that and pasted it and maybe crop it and just so I can enlarge it some and then maybe enlarge it. And you never know. I'm just going to see. Not that focused enlarged, okay? But still, that is something you can do. You really need to be stable, which is one thing about the high magnification in pictures. But honestly, for $20 instead of like thousands of dollars for a really nice microscope, for $20 and something you can use right at home, and this is pretty good. I mean, especially this is as good of an image as I have ever gotten at, usually it's I think it's 10x, but then 100 for total magnification or 400x. But this is as good as any image I've ever gotten on the onion skin, I believe. So you can do that and save them and label them if you want on here and save them and then as a whole image. Look, there's design ideas. So, and there is one other thing I would like to look at before we finish this. And let's see, share screen. And well, we got the cell lab. Let's see the answer sheet. So I'm just going to share the screen again. And I don't know why that thing is at the bottom. Answer sheet. It said see answer sheet. And it didn't open it. Answer sheet there. Make sure that I'm sharing this answer sheet. There. So on the answer sheet, it said see the parts to it. So questions about the magnification for the pocket microscope. So if you look at those links that I sent you, that should help. And oh, look, what do you add first to the slide when making the epithelial cell slide? Usually you add the methylene blue. But anyway, there you go. All of this, you can just follow. Oh, you're going to have to know which organisms are prokaryotic and which ones are eukaryotic. And you should know that. If you're not sure, you can go look at your, well, you can look it up on Google, but you can look at your background material notes. So here's where you can put the results. Again, if you can't get the images on here, which to get them on here, you could just go insert and then picture and find your pictures. I wonder if, no, that didn't work for me. I was going to say I had put one there earlier, but here's about the cells. Oh, label the diagram. I'll tell you what, a lot of people had really a problem with the diagram showing up from before, okay? From the lab, I don't remember which lab. Oh, bacteria and viruses were on it, the micro lab. I mean, it just didn't show up or getting the labels on or all of that. But if you want to just go in here and type in 1, 2, 3, 4, 5, 6, 7, 8, 9, what are there, 10 of them, and then just write what they are after it, then you can do that. At least you can write the number in there, so that may help. And then the same thing for this one. At least you don't have to put the words right on it, so it may be helpful there. Might just have to erase the image. And here's another one, the cell membrane. Excuse me, the cell membrane. The I'm not going to say what type of cell this is because then I would give it away. Well, if you watch the video this far and you're getting it, this is an animal cell right there. Give it away. It has no cell wall. It has a cell membrane, right? The plant cell has a cell membrane and a cell wall, okay? And it has chloroplast right here. And look at that giant vacuole. And this one right here, you should already know what type of cell this is from the microbiology module that we had last. So, and I believe this is it then. Yep, I believe this is it. So if you have any questions, email me. Good luck on this lab. And that's it. Have a good night or a good day.